Morning, class 8, ninth student. This is chapter 8, structure of seeds and seed germination. Okay, so there are varieties of seeds we generally consume in our daily life. So here you can see these are the learning objectives of this video lesson. Seed and types of seeds. So there are two categories in which the seeds can be divided. First one is endospermic and non-endospermic seeds. The second one is dicotyledonous and monocotyledonous seeds. Parts and structure of bean seeds and maize seeds, then differences between these two. Okay, so here you can see the structure, the general structure of a seed and embryo. As you know, embryo and endosperm, this I have already explained in the previous video lesson. This perisperm is new term for you. Just keep this perisperm term in mind. I'll explain later about this in this video lesson. Okay, so keep this in mind. Then the first topic of this chapter is seed. So seed is what? Seed is a ripened ovule, or we can say it is a mature ovule after fertilization. A seed contain embryo plant, stored food material. This stored food material can be in the form of cotyledons or in the form of endosperm. So embryo plant, then stored food material and seed coat. So you can see that this is embryo, this is stored food material, and this brownish structure is actually that seed coat. So this is seed, and the embryo remains in an inactive stage until exposed to favorable condition. This embryo remains inactive, or we can say in a dormancy stage, until it will get favorable condition. Favorable condition, it can be like water, warmth and air. Okay, so all this, when it gets all the necessary condition, then only the seed germinate. Okay, and you know what, that some seeds are known to remain dormant even up to 100 years. Yes, that for 100 years the seed, some of the seeds remain in dormancy. Okay, so this is about seeds. Now, types of seeds. So there are two divisions on which the seeds can be classified. The first category is endospermic and non-endospermic. So depending on the presence of endosperm, the seeds can be of two types, endospermic and non-endospermic. And depending on the number of cotyledons, the seeds can be divided into two, monocotyledonous and dicotyledonous. Okay, so let us discuss endospermic and non-endospermic seeds. So endospermic seeds and non-endospermic, it is very easy that endosperm, it means endospermic, that endosperm is present, and non-endospermic seeds, it means endosperm is absent. So now what is the role of endosperm? So if you remember in the previous video lesson, I told you that endosperm is what? That it provides nourishment to the developing embryo. Okay, so in the early early growth stage of embryo, the endosperm continues to store food, okay, and then develops more, which provide nourishment, to the developing embryo. And in this type of endospermic seeds, that cotyledon is very small, thin and papery. So this is the region where cotyledon is present, and this is very small, thin and papery, okay, or sometimes even absent also. So this is about endospermic seeds. This is endospermic seeds definition, and here example of endospermic seeds is given here. Okay, and another example of endospermic seeds can be like rice, uh, maize, or wheat. Okay, these are the example of endospermic seeds. Then non-endospermic seeds. So non-endospermic seeds, it means that endosperm is absent. So if it is absent, why it is absent actually? Because in the early stages, the endosperm is used by the embryo. Okay, in the early development stage, this endosperm is completely used up by the embryo. Then something should be there to provide food to the developing embryo, and that is cotyledons. So cotyledon is there which provides nourishment to the developing embryo. So here you can see this yellow part actually. So that this is called cotyledons. Okay, yes, this is cotyledon. So this cotyledon provides nourishment to the developing embryo, and it is example of non-endospermic seeds are gram, pea, beans, and pulses. So this is the picture of non-endospermic seeds, and this is the definition of non-endospermic seeds. Got it? The next category is monocots and dicot seeds. So monocot seeds, here you can see that depending on the number of cotyledons sometimes it is also called seed leaf. So inside the seed there is seed leaf also. So if the seeds have only one seed leaf, then we can say it is monocots. Okay. uh, or we can say that the seeds have one cotyledon, then it is called monocot. Example is rice, maize, wheat, onion. And dicots seeds have two cotyledons. Dicot seeds are far more in number than monocot seeds. Okay, so if it is one, then we can say monocots, and if it is more than one, it means that two, then we can say it is dicots. And monocot seeds are mostly endospermic in nature. It means what we have read in the previous video lesson, not in the previous, here in the previous slide, that endosperm. So monocot seeds are generally endospermic in nature, and dicots are generally non-endospermic in nature. Now, parts and structure of bean seeds. So here, the a mature seed contains two essential parts. This bean seed is non-endospermic in nature, and in your syllabus, two structures are explained. First one is bean seed, and second one is maize. So the maize seeds is divided into two categories. Has two parts, we can say, that seed coat and embryo. That seed coat is testa, tegumen, hilum, micropyle, raphe. All are actually comes under the category of seed coat. So that the structure of bean seed is divided into two. The first one is seed coat and the second one is embryo. And seed coat itself has this testa, tegumen, hilum and raphe, micropyle and raphe. Then embryo, which has cotyledons, radicle and plumule. Let us discuss about this and see what they are, what these parts are. So testa. Testa and tegumen are what? These are the outermost layer, or we can say these are the seed coats. So they are two layers. If you remember that integument, okay, that the mature ovule has integument. So these integuments are developed uh, into into testa and tegumen. Out of this two, which is the outermost? So testa is the outermost and toughest layer, a tough layer which does not allow entry of bacteria or any infection. Even that this testa also help in that protects the delicate inner parts of the seeds from injury, okay, and even from insects. And tegumen is the inner layer, okay, of the seed coat, and this is thin and inner layer and next to the testa, and it is also protective in nature. So both this layer which forms a seed coat, here you can see that brownish structure, this forms testa and tegumen. Outer layer is called testa and inner layer is called tegumen. Okay. Then next one is hilum. So hilum is what? Hilum is a, here you can see that hilum. It is a distinct oval scar on the concave side of the seed. This is a concave side, and here you can see a scar like structure is there. Okay, this hilum it represent the spot where the ovule was attached to the ovary. Ovule, this is ovule, okay, and when it is attached, when it was attached to the ovary, so it represents that point, okay, and through the placenta. Then the next one is micropyle. So micropyle, if you remember in the remember that what is micropyle? So it allows entry of pollen tube into the ovule. Yes, so the place which allows entry of pollen tube into the ovule, This is called micropyle. So for fertilization, it is really important, and for germination also this is important because it allows entry of water. It is a porous structure, okay, or a single pore which is present in the seed, okay. It allows entry of water, and also that it allows gaseous exchange or diffusion of gases, okay. And then refi is what? That refi is actually this portion, um, the line or that ridge like portion, that is called refi. And here hilum and micropyle are present in this refi, okay. So this overall what we have discussed, it is the part of seed coat. Now the next part is embryo. Embryo has three parts: cotyledon, radicle, and plumule. So cotyledon, you can see that the most part of the seed is cotyledon, okay. This is which helps to store food, okay, and provide it to embryo. Then the next part is called radicle. You can see this is the radicle part which undergoes development, and at the time of germination, after germination, it produces roots, okay. And the plumule part, here you can see that this is the plumule part which gives rise to shoot. It means it goes in upward direction, and this root will go in downward direction under the soil, okay. And there are two more structures you are seeing, hypocotyle and epicotyle. So epicotyle is upper stem and hypocotyle is lower stem. You just keep this thing in mind that radicle develops into root and plumule develops into shoot, okay. So this is the structure of bean seed. Now the next structure is structure of maize. So a mature seed contains three essential parts, and these are seed coat, endosperm, and embryo. All these are the this all these three are the essential part of the maize seed. This is a structure of maize seed. Let me explain it more to you. Seed coat. So seed coat is what? Here you can see there's a small structure. So this is a seed coat, okay. This is endosperm, and this is the embryo part. Seed coat is what? That actually, that about maize grain I want to tell you that maize grain is a small triangular grain. On one of its side is a small opaque whitish oval structure, okay. This oval area, the embryo is embedded in it. Here you can see this embryo is embedded in it, okay. In the oval structure, embryo is there. In a longitudinal section of maize, following parts can be seen. Let us see what these parts are. So first one is seed coat and pericarp. Yes. So seed coat or pericarp, what is it? Uh, the outer yellowish membranous layer is made up of seed coat. Here you can see this outer structure, okay. This outer yellowish structure, that is called uh, seed coat, and it is also have a pericarp. Pericarp, do you remember? I have explained in previous video lesson that what is it? Pericarp, if you remember that there are three layers in the pericarp. It is epicarp, mesocarp, and endocarp, about fruits, if you remember, okay. So yes, it is the same. So I can say about this maize grain that the maize grain is actually one seeded, and in which the fruit wall and the seed coat are fused together to form a protective layer. This two, that the seed coat and pericarp together form a protective layer. Therefore, we call such a fruit as grain. Okay, and the another structure, that is endosperm. So this part you are seeing, that is called endosperm, okay. So endosperm uh, is divided into two unequal portion. Here you can see unequal portion, and uh, it is actually that it is surrounded by epithelial, layer. Okay, epithelial layer. And the endosperms, the food material mainly in the form of starch. It stores food material in the form of starch, but here you can see that the line pattern. Okay, so that is actually another layer, and that is called alluron layer. And this alluron layer contains protein. Okay, so this from this part store food in the form of starch, and this alluron layer store food in the form of protein. Okay, so seed coat and endosperm is explained. Now the remaining part is about embryo. So cotyledon. Um, about embryo, I want to tell you that it is a lower side of the endosperm. Here it is present in the lower side of the endosperm and consists of single cotyledon, radicle, and plumule. Then now cotyledon. So cotyledon is what? The name of cotyledon is changed. It is a much reduced cotyledon known as scutellum. Scutellum is attached to the endosperm on one side by epithelium. है Here you can see that this is a epithelial layer. Okay, so on one side it is attached to epithelium. Its function is to digest and absorb. What is the function of this? So its function is to digest and absorb the food material which is provided by the endosperm. So and it takes the food material, digest it, and absorb it and make it available to this embryo, to the growing embryo. Okay, then the next part is plumule. So plumule, where it is present? It is present in the upper side of the embryo. Okay, in the upper side. Here you can say this is plumule. So it is present on the upper side of the embryo, and it is surrounded by a protective sheath, and that sheath is called coleoptile. So embryo is not just that plumule is there. It is also surrounded by a protective sheath, and that protective sheath is called coleoptile. And the next part is radicle, and radicle is what? It is present on the lower side of the embryo, and it is also protected by protective sheath, and that is called coleorhiza. Here you can see coleorhiza, which protects radicle, and plumule, which protects coleoptile. So this is an example of endospermic seeds in which scutellum, or we can say that coleoptile, is very small, thin, and papery. Okay. Now, the difference between dicot seeds and monocot seeds. So, what are the differences between these two types of seeds? Dicotyledonous seeds, that these are dicotyledonous, and monocot seeds, these are monocotyledonous. Dicot seeds are non-endospermic generally, and monocot seeds are generally endospermic. Seed coat is not fused with the fruit wall. The dicot seeds, that the fruit wall is not there, but we have discussed that in monocot seeds that the pericarp is there. Okay, and food is stored in two cotyledons. Yes, because they are dicot, it means two, so that two cotyledons are there. And monocot, it means that one cot or one cotyledon is there, so we know about this. Then food stored is mainly protein, may contain little starch fat. Such type of food material is proteinaceous. Okay, but such type of food materials, it is mainly that starch and some amount of the, because that alluron layer is there, so protein is also present. Hilum and micropyle are distinct on the surface. Yes, this is the hilum portion, and this is then here that micropyle is also there. We know that it is a porous structure. Okay, but such type of structure is not visible. Then develops in a pod or legume that has many seeds. Yes, so it develops into in pods or legumes which has so many seeds, and here it develops from a single seed fruit called caryopsis. So many maize grain together grow on a single fruit, or like that rice or that wheat, they grow together on one particular I can say fruit. Okay, so this is monocot seeds, that is called caryopsis. Such type of pattern is called caryopsis. Then plumule and radicle are not protected. Okay, but they are protected in monocots by sheath. And what the sheaths are called? coleoptile, and coleorhiza. Okay, so this are the differences between monocots and dicots. Now let us find the difference between endospermic and non-endospermic. This is very easy. That endospermic seeds are having endosperm. Endosperm is there, and non-endospermic seeds do not have endosperm. They have cotyledons. So here cotyledons, food is stored in that in non-endospermic seeds, food is stored in cotyledons, and here that food is not stored in cotyledons. The cotyledons in this category are small, thin, and papery, and here the cotyledons are thick and fleshy. Here endosperm is thick and fleshy. Okay, and as I told you that you have to remember one term, and that is perisperm. So perisperm does not develop from nucellus. In endospermic seeds, perisperm does not develop from nucellus. Nucellus, do you remember? Yes, that the multi-layered structure of the ovule. Okay, and here that perisperm develops from the nucellus. In non-endospermic seeds, the perisperm develops from the nucellus. So these are the differences between endospermic and non-endospermic seeds. Now this is a mini test for you. Okay, these are labelings. You have to label parts of seeds. I think you can do it. Okay, just after seeing this video lesson, after watching this video lesson, you have to try to do this. I think you can do. Okay, so have a nice day. Eat sprouted seeds, be fit and healthy, and keep studying. Bye.